for the kind introduction, and thanks for giving me the opportunity to showcase my research. Primary and metastatic brain tumors remain challenging to diagnose and treat due to the relentless tumor growth and invasion into normal tissue, which often goes unrecognized. For example, glioblastoma multiforme, or GBM, is the most common and fatal form of primary brain tumor. It has a peak incidence in adults older than 40 years of age, with roughly 3 cases per 100,000 people annually in North America. Current standard care of treatment for GBM is a surgical resection, followed by fractionated radiation and chemotherapy. And patients diagnosed with GBM have a mean survival period of only 12 to 15 months, with only 10% surviving 5 years. Contrast-enhanced T1 and T2 weighted MRIs are commonly used to identify brain tumors. However, conventional MRI of gliomas has inherent limitations, such as inadequate assessment of peritumoral edema and integrity of adjacent white matter. Microinvasive tumor cells, which has not been detected on conventional MR imaging, contribute to poor prognosis for patients diagnosed with GBM. But on the other hand, diffusion-weighted MRI methods are being developed to detect the presence of tumor boundaries that may be different from those seen by more conventional methods. Diffusion-weighted MRI allows non-invasive measurement of translational motion of water in living tissue, and it is sensitive to tissue microstructure. When water molecules diffuse freely, such as in a glass or a brain ventricle containing cerebrospinal fluid or CSF, the random displacement of water molecules obey Gaussian distribution. In tissue, diffusion is constrained by molecular or cellular obstacles. So displacement distribution becomes sharper, especially in certain pathologies, such as brain tumors with high cell density. So characterizing the diffusion of water within and surrounding the tumor may help to identify the extent of cancerous tissue. A more advanced diffusion-based MRI method is diffusion tensor imaging, or DTI. In DTI, once the water diffusion is measured, diffusion in each voxel is modeled with diffusion tensor, which can represent as an ellipsoid with 3 eigenvalues, eigenvectors, and corresponding eigenvalues. We obtained tensor-based metrics from these tensor to describe the diffusion within each voxel. These measures are pretty sensitive to some tissue properties, such as axonal density, cellularity, inflammation, and degree of myelination. In DTI, 4 measures are most used: axial diffusivity, radial diffusivity, mean diffusivity, and fractional anisotropy. If the presence of cancerous cells changes the macroscopic organization of a surrounding cell, it is possible that diffusion properties of water within the tumor and its surrounding will be altered. So this study aimed to characterize the tissue microstructure differences between glioma tissue and healthy brain tissue in rat using diffusion tensor imaging at 9.4 Tesla MRI. So we hypothesized that there are distinct patterns of water diffusion with a tumor compared to its surrounding and healthy brain tissue. For this study, we used C6 glioma cells, and we manually injected those cells into the right brain hemisphere of 6 8-week-old male rats. The C6 cell line overexpresses several genes similar to those reported for human gliomas. So that's why we chose this cell line. Tumor-bearing rats underwent imaging using 9.4 Tesla on day 14 following tumor implantation. And diffusion tensor imaging were acquired using a sphenoid DI diffusion rated sequence. 100 diffusion rated images with different p-values were acquired, and images were pre-processed to correct susceptibility and eddy current induced distortion. Then DTI metrics were generated. The same procedure was applied to the 6 healthy control male rats. Okay. In this figure, the top row is T2 weighted images of a tumor-bearing rat, like a first image, and the rest of the images are DTI metrics for the same rat 14 days following implantation. And the DTI metrics shown here are fractional anisotropy or FA, mean diffusivity or MD, axial diffusivity or AD, and radial diffusivity or RD. I outlined the tumor region in purple on T2 weighted image. By day 14, an increase in signal intensity is observable in T2 weighted image. And tumor mass effect is apparent as the ventricular space and white matter tracts are displaced. You can see the same images for a healthy control rat brain in the bottom row, like anatomical, and the rest are DTI metrics. Different contrasts represent different tissue. For example, in a FA map, white matter tracts are bright and gray matter and CSF are not. Fractional anisotropy measure macrostructural integrity and is highly sensitive to their changes. Mean diffusivity is sensitive to CSF, which has high value of average diffusion. And it's sensitive, at the mean diffuse, it's sensitive to cellularity, edema, and necrosis. Axial diffusivity tend to be variable in white matter changes and pathology. Radial diffusivity increases in white matter with demyelination or dysmyelination. Also, changes in axonal diameter or axonal density may influence radial diffusivity. For our analysis, we manually do region of interest on FA maps. The region of interest that for tumor-bearing rats are tumor in yellow, tumor rim in blue, and contralateral gray matter in pink. For the healthy control rats, region of interest are right and left side gray matters. We use pair t-test for our stat analysis to compare DTI indices between ROIs. Here are the quantitative analysis of DTI indices between ROIs in tumor-bearing rats. Tumor versus contralateral gray matter is in figure A, and tumor versus tumor rim in figure. Back to figure A. We found a significantly higher FA, MD, axial diffusivity, and radial diffusivity in tumor region compared to contralateral gray matter. Our result is consistent with DTI findings in similar tumor model. Peritumoral edema can be classified as compressed normal tissue, pathogenic edema, and tumor cell infiltration edema. The ability of DTI to differentiate those is still controversial. That's why I call this region rim. So, and we need histology to confirm if the rim is edema or not. And the tumor ring was defined as a bright ring surrounding the tumor. In figure B, significantly higher FA, mean diffusivity, and radial diffusivity were found in tumor region compared to the tumor ring. Here are the quantitative analysis of DTI indices between tumor-bearing rats and healthy control rats. As you see in figure A, significantly higher mean diffusivity, axial diffusivity, and radial diffusivity they're found in tumor and gray matter area of the healthy control animals. However, only the average average FA values significantly differ between contralateral gray matter and healthy control gray matter, as shown in figure B. In figure C, we observe that significantly higher FA, mean diffusivity, and axial diffusivity values between tumor rim and healthy control gray matter. So, as expected, noticeable changes in water diffusion properties within and surrounding the tumor were observed. Cystic glioma cell is an aggressive tumor model. So we propose that the rapid growth in this tumor model exert pressure on surrounding tissue. So we can say those observed differences in diffusion metrics may be related to increasing cellularity or changes in cell geometry in tumor region. Future work will be involved in correlation study of DTI and histology, and also we are interested in comparison between DTI and myometrics. With that, I would like to acknowledge my supervisor, colleagues, and funding sources. And thank you all for listening, and I will be happy to take any questions.